Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and we are into the revision mode and we are revising chapter number 11, Biotechnology, Principles and Processes, and this is meant for 12th November 2020. As we have been talking about the importance of this chapter, and there are two chapters, biotechnology chapters, and in this unit these two chapters are very important as far as your CBSE board exam is concerned, and also lot many questions in your NEET exam. Plus in future, as I have been telling you, this will be a bread winner for you. You may become a biotechnologist in future, and this gives a career for you. Hence, focus on this chapter. So what is biotechnology? Biotechnology is the use of living organisms in systems or processes for the manufacture of useful products. It may involve algae, bacteria, fungi, yeast, or cells of higher plants and animals, or subsystems of any of these, or isolated components from living matter. The term was given by Hungarian engineer Carl Erki in 1917. So the techniques of using live organisms or the products, enzymes, from organisms to produce products and processes that are useful to humans. Many processes like in vitro fertilization leading to test tube baby, synthesizing gene and using it, developing a DNA vaccine or correcting a defective gene are also parts of biotechnology. The European Federation of Biotechnology has given a definition of biotechnology that comprises both traditional and modern molecular biotechnology. The definition is as follows: the integration of natural science and organisms, cells, parts thereof, and molecular analogues for products and services. So basically, the science of biotechnology is based on two core technologies or two core principles, and the modern biotechnology is always based on two technologies and two principles. One is genetic engineering and the second one is biochemical engineering. So what is genetic engineering? Genetic engineering is defined as the direct manipulation of genome, means set of all the genes, DNA and RNA of an organism. So it is a manipulation of DNA and RNA. It involves the transfer of new genes to improve the function or trait means character, into host organisms, and thus changes the phenotype. Phenotype means physical appearance of an individual, and this phenotype, physical appearance of an individual, is due to its genotype and the phenotype of the host organism. Maintenance of sterile condition in chemical engineering processes to enable growth of only desired microbes for manufacture of biotechnological products like antibiotics, vaccines and enzymes. This is biochemical engineering. So genetic engineering refers to artificial synthesis, isolation, modification, combination, addition and repair of DNA by man to alter the phenotype of the host organism to suit our needs, that is human needs. It includes formation of rDNA, recombinant DNA. So basically in genetic engineering what we are doing is we are recombining the DNA. So recombinant DNA, making a rDNA is the main target in genetic engineering. And this recombinant use of gene cloning and gene transfer takes place. And what about biochemical engineering? It involves processes that help the growth of desired microbes, eukaryotic cells in large quantities in a sterile medium, that is tissue culture technique, for the manufacture and multiplication of biotechnological products such as antibiotics, vaccines, enzymes, medicines and hormones. So the two principles on which the biotechnology is based is, one is the genetic engineering, we will write it as GE, and another is biochemical engineering. Second principle is biochemical engineering. This is a new branch of engineering, biochemical engineering. Okay, very few universities offer this, so you need to go to European countries for this. And traditional hybridization used in plants and animals breeding leads to inclusion and multiplication of undesirable genes that we don't want, along with the desired traits. The technique of genetic engineering, which include creation of rDNA, use of gene cloning and gene transfer, allow us to isolate and introduce only one or a set of desirable genes without introducing undesirable genes. This is our target into our target organism. So in a chromosome there is a specific DNA sequence called the origin of replication. We say ORI, origin of replication, which is responsible for initiating replication. Replication is DNA division. Therefore, for the multiplication of any alien piece of DNA in an organism, it needs to be part of a chromosome which has specific sequence known as origin of replication. Thus, an alien DNA is linked with the origin of replication so that this alien piece of DNA can replicate and multiply itself into the host organism. This is known as cloning, or making multiple identical copies of any template DNA. This is the secret behind it. The construction of the first rDNA emerged from the possibility of linking a gene coding or encoding antibiotic resistance with a native plasmid of Salmonella typhi. Stanley Cohen, Herbert Boyer. So Cohen and Boyer in 1972 isolated the antibiotic resistance gene by cutting out a piece of DNA from a plasmid. What is plasmid? Extra chromosomal circular DNA. So autonomously replicates this itself replicates. So autonomously replicating circular extra chromosomal DNA is plasmid, and this is of Salmonella typhi bacterium. The cutting of DNA at specific locations became possible with the discovery of so-called molecular scissors. And what are these molecular scissors? Restriction enzymes. So the cut piece of DNA was then linked with the plasmid DNA. So you got a cut piece of DNA and then you got a plasmid DNA. So two are there. These plasmid DNA act as vectors to transfer the piece of DNA attached to it. So this piece is attached to it. So as a result, this piece which we are attaching, desirable one, we can put this into the host, into a bacteria, virus, and allow it to multiply. So these vectors to transfer the piece of DNA attached to it. A plasmid can be used as vector to deliver an alien piece of DNA into the host. So this is itself can't go into a host, but when it is attached here to the plasmid, the entire plasmid can be made into the host. Then it gets multiplied. So the linking of antibiotic resistance gene with the plasmid vector became possible with the enzyme ligase or ligase, which acts on cut DNA molecules. And what is the function of ligase? Joins their ends, joining. This makes a new combination of autonomously replicating DNA created in vitro and known as rDNA. Now we call this as recombinant DNA. When this DNA is transferred into E. coli, it could replicate using the new host DNA polymerase enzyme and make multiple copies. The ability to multiply copies of antibiotic resistance gene in E. coli was called cloning of antibiotic resistance gene in E. coli. So rDNA technology, also called genetic engineering, deals about the production of new combinations of genetic material in the laboratory. These rDNA molecules are then introduced into host cells where they can be propagated and multiplied. So basically, the tools for recombinant DNA technology, we need first enzymes, okay, and after enzymes we require the separation, the gel electrolysis and the cloning vectors. Then we require competent host, and then of course we go for the processes, steps in recombinant DNA. So basically, identification of DNA with the desirable genes, the first step. Second, introduction of the identified DNA into the host, second. And then the third one, maintenance of introduced DNA into the host and transfer of the DNA to its progeny. These are the three main steps. And what are the tools we require? As I said, first we require restriction enzymes, then to expand that, polymerase enzymes, then to join the pieces, ligases, then we require a vector, and of course a suitable host. And when these tools are available to us, it is easy for us to do the genetic engineering. Restriction enzymes, they're called molecular scissors, as we said just now. Restriction enzymes belong to a larger class of enzymes called nucleases. There are two kinds, exonucleases, endonucleases. As the name says, exo means outside, periphery, endo means inside. Exonucleases remove nucleotides from the ends of DNA, only the ends it will remove, whereas endonucleases make cuts at a specific position within DNA. That's why endonucleases are very, very important. Example, the first restriction endonuclease Hind2 always cut DNA molecule at a particular point by recognizing specific sequence of 6 base pairs. This specific base sequence is known as recognition sequence for Hind2. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence in the DNA. Palindromes are a group of letters that form the same words when read both forward and backward. For example, Malayalam, like M-A-L-Y-A-L-A-M. Reverse it, M-A-L-A-Y-L-A-M. So this is similarly a very familiar one, Gadag, G-A-D-A-G, G-A-D-A-G and G-A-D-A-G. Such things are called palindromes. So you can see here 5' to 3', G-A-A-T-T-C. What are these G-A-A-T-C? Nitrogen bases. Guanine, adenine, adenine, thymine, thymine, cytosine. And reverse of it, this way, G to C, now G to C. Then we say palindrome. So the palindrome in DNA is a sequence of base pairs that reads same on two strands when orientation of reading is kept the same. So this is how the endonucleases cut the pieces, as you are seeing here. The enzyme cuts both the DNA strands at the same site. You can see here vector DNA, foreign DNA. Now there is a cut portion, sticky ends. And these sticky ends now joined and rDNA is formed. So this is how rDNA can be made. So keep practicing the diagrams, label it, understand the